Hello students, Class 8 AB Biology, Chapter 2 Reproduction in Plants and Animals, Part 1. Show the following processes in the form of flowcharts. Binary fission. Nucleus first divides into two nuclei. Cytoplasm divides into two parts each containing a nucleus. Two identical cells called daughter cells are formed. Tissue culture. The shoot tip is cut from the main plant and divided into many small pieces. Shoot tip pieces kept in the nutrient medium in a test tube. Tissues grows into callus. Callus in presence of growth hormone grows into the plantlet which ultimately divides into new plant. Number 3. Fertilization in human. The sperm that enters the vagina swim upwards. Many sperms approach the egg. Only one sperm enters. Nucleus of the sperm approaches the nucleus of the egg. Other sperms die and disintegrates. Nuclei of sperm and egg unite to form zygote. The zygote moves from the oviduct to the uterus where further development takes place. Number B. Give reasons for the following statements. Vegetative propagation is common in plants that do not produce seeds. Vegetative propagation is common in plants that do not produce seeds because in such plants, development of a new plant takes place from the vegetative parts like root, stem, etc. Number 2. A very large number of sperms are present in the semen. Sperms are produced in large numbers to ensure the fertilization of the ovum. Flowers that are pollinated by wind have large stigma. Flowers that are pollinated by wind have a large stigma to receive pollen grains. Zygote is the basic unit of life. Zygote is the basic unit of life because it contains the genetic material of both the mother and the father and after cell division becomes an embryo. Answer in short. What is pollination? Name the agents of pollination. Pollination is the transfer of pollen grains from anther of a flower to stigma of the same or another flower. The agents of pollination are wind, insect, water and animals. 2. Give any two advantages of vegetative propagation. Any two advantages of vegetative propagation are as follows. Flowers produced are of superior quality. Plants that do not have viable seeds can be reproduced. Give any two characteristics of insect pollinated flowers. Any two characteristics of insect pollinated flowers are they are large and bright colored, they have sweet nectar and are scented. 4. What is the benefit of artificial pollination? The benefit of artificial pollination is that it develops a variety of crop having characteristics of two plants. What is semen? What does it consist of? Semen is the mixture of sperms and secretions from seminal vesicles, prostate, corpus and urethral glands. Semen is made up of sperm cells. Define the term ovulation. Ovulation is the process of release of an egg by the embryo into the fallopian tube. At length, explain the following terms. Reproduction. The process by which living organisms produce new individuals of their kind is called reproduction. Pollination. The transfer of pollen grains from anther of a flower to stigma of the same or another flower is called pollination. Hybridization. Crossbreeding done by artificial pollination to develop a new variety of crop is called hybridization. Gynoecium, it is the female reproductive part of the flower and consists of one or more pistils or carpels. Unisexual flowers, the flowers which contain only the male or female reproductive organs are called unisexual flowers. Answer in detail 1. What is vegetative propagation? How does it take place? The development of a new plant from the vegetative parts like root, stem or leaf of a plant is called vegetative propagation. It takes place by natural or artificial methods. In natural method, vegetative propagation takes place naturally from different parts like stem, leaves and roots of the plant. Whereas in artificial method, Vegetative propagation is carried out by human beings to grow plants. Number 2. Explain the process of tissue culture. What are its advantages? In the method of tissue culture, at first, the shoot tip is cut from the main plant and is then divided into small pieces of tissues. Each piece is then put in the nutrient medium in a test tube and grows in an unorganized mass called callus. Small parts of callus are transferred into another medium containing growth hormones which induce the formation of plantlets. These plantlets are transferred to the soil and ultimately develop into new plants. Advantages of tissue culture are as follows. The new plantlets can be grown in a short amount of time. The new plantlets and plants are more likely to be free of viruses and diseases. The process is not dependent on the season and can be done throughout the year. Now here we will draw the tissue culture image. It's given in page number 22, figure 2.7. Thank you.